Hello, good morning students. Welcome to the Gnan Sagar video lecture series. I hope you enjoyed the first two lectures of biology and I hope you also studied the contents of the two lectures from the videos and from the NCERT book as well. Now today we will discuss the next point in the third video lecture, that is the DNA molecule. If you look at this chapter in your NCERT, this point is given as a first point in the NCERT, and then they have given as a second point the search for genetic material. But I think that sequence is a little bit wrong. First we should clarify what is the genetic material, and once the genetic material is cleared in your mind, it is DNA due to these things, proven due to these experiments, then we should study the detailed information about the genetic material. In this chapter we will study the structure, the dimensions of the DNA molecule, the packaging of the DNA molecule in the chromosomes, and then we will study the metabolism of the DNA molecule. For example, its replication, its transcription and production of RNA, and then further translation and production of proteins from the RNA. So today we will talk about the dimensions and the structure of the DNA. Now DNA was first identified as an acidic substance by Friedrich Mischer in the nucleus of eukaryotic cells, and he named that substance, acidic substance in the nucleus, as nuclein, as it was found in the nucleus of the eukaryotic cells. The length of the DNA is expressed either in nucleotides, number of nucleotides, or in number of base pairs, for example as written here, and that depends whether the DNA molecule which is present as a genetic material of an organism or of the virus is single stranded or double stranded. In almost all the cellular forms, all the living organisms, the genetic material is double stranded DNA. In some of the viruses also the genetic material in the form of DNA is double stranded, but in some viruses the genetic material is single stranded DNA, and as we also studied in the last lecture, in very few viruses the genetic material can be RNA also. The examples of RNA as genetic material for some viruses are tobacco mosaic virus in plants, then HIV virus in humans, and this coronavirus, the outbreak of which we are facing today, this is also having RNA as a genetic material. So when single-stranded RNA or single-stranded DNA is the genetic material of any virus, then we refer its length in number of nucleotides, and if the genetic material is double-stranded in virus or in any organism, in bacteria or in higher organisms, then it is referred in base pairs. So now for example, the length of bacteriophage 5x174 DNA, which is a single-stranded DNA, is 5386 nucleotides, referred in nucleotides because the genetic material is single-stranded DNA. And for all other some viruses and some organisms, the DNA is double-stranded. For example, bacteriophage lambda, which infects E. coli, has 48,502 base pairs as its length of the DNA, and the base pairs indicate that it is a double-stranded DNA, and as an additional information I will say that it is a circular DNA inside the bacterial cell, but it is a linear DNA inside the capsule of the bacteriophage lambda. How it assumes the circular nature inside the bacterial cell, that we will discuss in biotechnology chapter. So this length 48,502 base pairs can be also expressed as 48.5 kilo base pairs or kbps. E. coli DNA length is 4.6 into 10 raised to 6 base pairs. E. coli has a single double-stranded DNA, circular DNA, as its chromosome, and the chromosome contains 4.6 into 10 raised to 6, 4.6 million base pairs. And human haploid DNA content, which is made up of 23 chromosomes, has 3.3 into 10 raised to 9 base pairs as its length. That means 3.3 billion base pairs. Now this is about the length of the DNA in general, but the most important factor that we have to study in this point is the structure of DNA. Now DNA is a polymer of deoxyribonucleotides, and it is therefore called as polynucleotide chain, a chain of several nucleotides. And each nucleotide is made up of three components. The first component is a nitrogenous base, the second one is a pentose sugar, and the third one is the phosphate group. The same three components are present in the nucleotides of DNA and in the nucleotides of RNA. The nature of the pentose sugar in the RNA and DNA differs, and one of the nitrogenous base in DNA and RNA differs. इतना ही difference है DNA and RNA के structure में. Otherwise, DNA and RNA both contain the same types of phosphate groups, three common nitrogenous bases as adenine, guanine, and cytosine, and the pentose sugar is slightly different in the RNA and DNA. Now we will first look at the nitrogenous bases which are present in DNA and in RNA as well. The nitrogenous bases are broadly categorized into two categories. The first one is purines and the second one are pyrimidines. The purines are double ring structures. The pyrimidines are single ring structures. There are double bonds inside the rings also, but I haven't shown them here. Purines are of two different types, adenines and guanines, and pyrimidines are of three different types, cytosine, thymine, and uracil. Out of these five nitrogenous bases, adenine, guanine, cytosine, and thymine, these four nitrogenous bases, they are present in DNA, and in RNA also adenine, guanine, and cytosine are present as common nitrogenous bases with DNA, but RNA does not contain thymine. Instead of thymine, RNA contains uracil, and DNA never contains uracil. It always contains thymine instead of uracil. Now we will see the next part of the structure of DNA. Okay, so in the first part we studied the structures of nitrogenous bases or types of nitrogenous bases. Now we will discuss about the structures of pentose sugars which are present in DNA and in RNA. So the type of pentose sugar that is present in RNA is called as ribose sugar, and its variant which is present in DNA is called as deoxyribose sugar. The reason for name deoxyribose is that from the second carbon, this is the first carbon of the sugar, this is second carbon, third, fourth, and fifth. So from the second carbon of the ribose sugar, one oxygen is missing in the deoxyribose sugar. So एक ऑक्सीजन यहाँ से रिमूव हो गया, निकल गया, इसलिए इसको deoxyribose शुगर बोलते हैं. तो ये राइबोज शुगर तो है ही, इसमें से एक ऑक्सीजन सिर्फ कम है. तो डीएनए के अंदर डीऑक्सीराइबोज शुगर होता है, इसलिए इसका नाम भी डीऑक्सीराइबोज न्यूक्लिक एसिड होता है. ये भी न्यूक्लिक एसिड है, पर इसमें डीऑक्सीराइबोज शुगर नहीं है, ऑक्सीराइबोज यानी सिंपली राइबोज शुगर है, इसलिए इसको राइबोज न्यूक्लिक एसिड ऐसा कहते हैं. तो दिस इज द सेकंड कंपोनेंट ऑफ एनी न्यूक्लियोटाइड, ribonucleotide or deoxyribonucleotide. The third component of the nucleotide is phosphate group. Now we will see how the nucleotides are exactly synthesized. The nucleotides are not directly synthesized from the three components. The first two components, the pentose sugar and the nitrogenous base, they combine with each other and they form nucleosides. For example, in this case, we are taking thymine as a nitrogenous base, which is a pyrimidine single ring structure, and this is a deoxyribose sugar. So they are not linked with each other in this diagram, but when they combine with each other, a condensation reaction occurs between this hydroxyl group and this hydrogen, so or between the first carbon and the nitrogen of the thymine or the nitrogenous base. तो यहाँ से ये H2O के फॉर्म में OH and H निकल जाते हैं, and there is formation of a bond between the first carbon of the pentose sugar and the nitrogen, first nitrogen of the nitrogenous base here, thymine. So due to that condensation reaction, this bond is formed between this carbon and this nitrogen, so that the nitrogenous base is linked with the pentose sugar, and this bond is called as a glycosidic bond. Glycosidic indicates that it is a bond with a carbohydrate. So the carbohydrate is forming a bond with the nitrogen of the nitrogenous base. So because nitrogen is involved in the bond formation, it is called as N-glycosidic bond. There are two types of glycosidic bonds, N-glycosidic bond and O-glycosidic bond. If oxygen is the intermediate in the bond formation, glycosidic bond formation, then that is called as O-linked bond, glycosidic bond. And if nitrogen is present or as a part of the glycosidic bond, then it is called as N-glycosidic bond. एग्जाम में जनरली इसके ऊपर क्वेश्चन पूछा जाता है कि नाइट्रोजनस बेस पेंटोज शुगर से कौन से बॉन्ड से लिंक्ड होता है, तो यू हैव टू रिमेंबर नॉट ओनली द ग्लाइकोसाइडिक बॉन्ड बट द एन ग्लाइकोसाइडिक बॉन्ड. तो इस तरीके से जब कोई भी नाइट्रोजनस बेस पेंटोज शुगर के साथ कंबाइन होता है तो न्यूक्लियोसाइड बनता है. तो यहाँ पे नाइट्रोजनस बेस थायमीन है, पेंटोज शुगर डीऑक्सीराइबोज शुगर है, इसलिए ये थायमिडीन वाला न्यूक्लियोसाइड बन गया. ओके, इट इज थायमिडीन. एंड इसी तरीके से अगर एडेनाइन यहाँ पे नाइट्रोजनस बेस है तो दैट विल बी ए डीऑक्सी न्यूक्लियोसाइड ऑफ एडेनाइन विच विल बी कॉल्ड एज डीऑक्सी एडेनोसाइन. यहाँ पे इसको कहेंगे डीऑक्सी थायमिडीन. अगर साइटोसिन यहाँ पे नाइट्रोजनस बेस है देन इट विल बी रेफर्ड एज डीऑक्सी साइटिडीन. एंड इफ there is guanine as a nitrogenous base which is attached with the pentose sugar by the N-glycosidic bond, then we call the nucleoside as the guanosine or deoxyguanosine. And if RNA is present, मतलब pentose sugar in this case is not deoxyribose but it is ribose, then all these nucleosides will be ribonucleosides instead of deoxyribonucleosides. And now we will discuss about the formation of a nucleotide from nucleoside and the third component, that is phosphate group. So a nucleoside is already shown here which forms in this fashion, and the third component joins with the fifth carbon of the pentose sugar by another condensation reaction. So what happens here, when phosphate group comes close to the fifth carbon of the pentose sugar, then again the hydroxyl group of this CH2 fifth carbon and hydroxyl group of the phosphate, they get involved in the condensation reaction, and one water molecule is lost between them, and this fifth carbon is linked directly with the phosphate of the phosphate group, so that a bond which involves oxygen as an intermediate is formed, and that bond is called as phosphoester bond. Remember, this is not phosphodiester bond, this is just a phosphoester bond. When two phosphoester bonds involve in the joining of two nucleotides, then the bond is called as phosphodiester. But here two nucleotides are not linked with each other. This is just a formation of the nucleotide which involves only one ester bond, and the ester bond is having phosphate along with it. That is why it is a phosphoester bond. So the formation of nucleoside involves N-glycosidic bond between the nitrogenous base and the pentose sugar, and the formation of nucleotide involves formation of a phosphoester bond between the fifth carbon of the pentose sugar and the phosphate group. तो अब ये थायमीन का न्यूक्लियोटाइड है, डीऑक्सीराइबोज शुगर है, इसलिए हम इसको कहेंगे थाइमिडिलिक एसिड और डीऑक्सीथाइमिडिलिक एसिड. अगर यहाँ पे नाइट्रोजनस बेस एडेनाइन है, then the nucleotide will be referred as deoxyadenylate acid. Likewise, deoxyguanylic acid if there is guanine, deoxycytidinic acid if there is cytidine. And in the RNA, the pentose sugar will be ribose, and in that case, the nucleotides will be referred as uridylic acids, adenylic acids. No deoxy word will be used in that case, and there will be no thymidilic acid because uracil is present in the RNA instead of thymine. Thymine is never present in the RNA. So in this fashion, due to the formation of specific bonds between the nitrogenous bases and sugar, and between the phosphate group and the sugar, a nucleotide is formed, and then several nucleotides can be linked with each other. If two nucleotides get linked with each other, we form a dinucleotide with the help of a single phosphodiester bond, and in this way several nucleotides can get linked with each other. We will see how the polynucleotide chain forms then. Okay, so we studied different components of the nucleotides. Now we will study how a phosphodiester bond forms between two nucleotides and how then a polynucleotide chain is synthesized. Suppose this is the first nucleotide at the 5' end of the polynucleotide chain, and this is the second nucleotide which is trying to get attached with the 3' end of the first nucleotide. So these two nucleotides come with each other, and again one dehydration or condensation reaction occurs between the two. This hydroxyl group of the second nucleotide's phosphate group and the hydroxyl group present on the third carbon of the first nucleotide's pentose sugar, they condense with each other, and in a dehydration reaction one water molecule is lost. तो ये water molecule lose होने के वजह से ये 3' कार्बन इस ऑक्सीजन के थ्रू इस फॉस्फेट ग्रुप के साथ ज्वाइन हो जाता है. तो यहाँ पे और एक फॉस्फोईस्टर बॉन्ड बन गया, कार्बन लिंक्ड विद द फॉस्फेट वाया ऑक्सीजन. यहाँ पे ऑलरेडी एक फॉस्फोईस्टर बॉन्ड बना हुआ था, कार्बन लिंक्ड विद द फॉस्फेट वाया द ऑक्सीजन. सो ये एक फॉस्फोईस्टर बॉन्ड, ये एक फॉस्फोईस्टर बॉन्ड, इस तरीके से दो फॉस्फोईस्टर बॉन्ड्स दो न्यूक्लियोटाइड्स को एक दूसरे के साथ ज्वाइन कर रहे हैं. दैट इज व्हाई दिस बॉन्ड combinedly is called as phosphodiester bond because there are two ester bonds. तो इस तरीके से फर्स्ट न्यूक्लियोटाइड इज लिंक्ड विद द सेकंड न्यूक्लियोटाइड, एंड इसी फैशन में इन द सेम वे third nucleotide will be linked with the 3' carbon of the second nucleotide, then fourth nucleotide's 5' end will be linked with the 3' end of the third nucleotide, and a long chain of nucleotides or a polymer of nucleotides will be formed, and that polymer of nucleotides will be called as polynucleotide chain, or it will be also called as deoxyribonucleic acid. तो इस तरीके से 5' end फर्स्ट न्यूक्लियोटाइड का हमेशा फ्री रहता है इन ए पोलीन्यूक्लियोटाइड चेन, एंड 3' hydroxyl group of the last nucleotide is always free in the polynucleotide chain. This is how a polynucleotide chain forms. Now we will discuss the double stranded structure or double helical structure of the DNA. Okay, now we will study the double helical structure of DNA, which was proposed by James Watson and Francis Crick, and they used the X-ray diffraction data proposed by or produced by Maurice Wilkins and Rosalind Franklin to generate the structure, double helical structure of DNA. They also were helped by the observations of Erwin Chargaff and the rule of Erwin Chargaff for the double-stranded DNA. We will talk about that rule later on, but we will first see the structure which was proposed by Watson and Crick, the DNA double helical structure. So according to the double helical structure model, the DNA is made up of two strands, and the two strands are coiled over each other in helical fashion. That is why, because there are two strands of DNA, it is double, and the two strands are present in a helical form coiled over each other. That is why it is called as double helical structure of DNA. So in the double helical structure of DNA, the two strands don't run parallel to each other, but they run anti-parallel to each other. मतलब the DNA synthesis always starts from 5' end and it ends at the 3' end. If one strand is running from top to bottom 5' to 3', the other strand always runs from bottom to top from 5' to 3' end. तो दोनों strands एक ही डायरेक्शन में 5' to 3' नहीं रन कर रहे हैं, एक 5 प्राइम टू 3 प्राइम इस डायरेक्शन में जा रहा है तो दूसरा 5 प्राइम टू 3 प्राइम अपोजिट डायरेक्शन में आ रहा है, एंड दैट इज व्हाई द टू हेलाइसेस are the two helices or the two strands, they are anti-parallel to each other. Another component of the double helical structure of DNA is that one turn of any strand over the other strand, मतलब complete 360 degree turn of one strand over the other strand is of 3.4 nanometer length, which can be also said as 34 angstrom unit, and each turn contains approximately 10 base pairs. One base of one strand pairs with the other base of the other strand. G always pairs with the C and T always pairs with A according to the Chargaff's rule. So this base pairing is present in the double stranded DNA with the help of which the two strands are held together. The base pairing is present between the opposite bases with the help of hydrogen bonds. So guanine base pairs with cytosine with the help of 3 hydrogen bonds, and adenine base pairs with thymine with the help of 2 hydrogen bonds. Adenine always base pairs with thymine and guanine always base pairs with cytosine in any double stranded nucleic acid. And these observations were given by Chargaff. Chargaff actually took double-stranded DNA in a test tube and he analyzed the components of the double-stranded DNA by degrading the DNA. तो किसी भी double-stranded DNA को degrade करने के बाद और सारे phosphodiesterase bonds cleave करने के बाद उसका analysis किया जाए तो हमको ये पता चलता है कि उस double-stranded DNA में जितने adenine है exactly उतने ही thymine है. That is why A to T ratio always is same and it is 1. जितने ग्वानाइन किसी भी डबल स्ट्रैंडेड डीएनए में है उतने ही साइटोसिन्स होते हैं. दैट इज व्हाई G to C ratio is also equal to 1. That means A's are always equal to T's and G's are always equal to C's. That is why A plus G upon C plus T comes to 1. That means all the purines which are present in double stranded DNA are equal in number to all the pyrimidines that are present in the double stranded DNA, which can also be stated like this: purines is equal to pyrimidines, or purine by pyrimidines is equal to 1. So this observation of the Erwin Chargaff also helped Watson and Crick to propose this double-stranded structure of DNA. Now the third aspect of the double helical structure is that the width of the double helical structure is uniform and it is 2 nanometer. It is uniform throughout. In the board, in the diagram which is written on the board, we don't see the uniformity because we are looking at the groove here, but actually in the 3-dimensional structure this is not a narrow groove, but the structure is ladder like, and the width of the ladder is always uniform from top to bottom. Likewise the width of this ladder is also uniform, which is 2 nanometer or 20 angstrom unit. And the reason for constant width is that a purine always base pairs with pyrimidine. You can think it in this way: a purine is a double ring structure like this, and in front of a purine there is always a pyrimidine which is a single ring structure. If the purine is guanine, the pyrimidine in front of it is cytosine. If the purine is adenine, the pyrimidine in front of it is always thymine. And because a purine is always present along with a pyrimidine, there is a space of 3 rings in between the backbone of the 2 strands. So if a purine base pairs with purine, then there will be 4 rings and the structure of the width of the DNA will extend. If 2 pyrimidines are present in front of each other, for example like this, they are only single ring structures, then in that case the width of the DNA will decrease. So if 2 purines come in front of each other, the width of the DNA will increase. 2 pyrimidines come in front of each other, the width of the DNA will decrease. And it never happens in the DNA. In the DNA always a pyrimidine is present in front of a purine, and that is why the width is constant 2 nanometer throughout the length of the DNA. Third aspect. The fourth aspect is that the distance between every two base pairs is always 0.34 nanometer or 3.4 angstrom minute, and that can be calculated because one turn contains 10 base pairs and the length of the total turn is 3.4 nanometer. So 10 base pairs are present in each turn, so the distance between every two base pairs will come out to be 3.4 nanometer or 0.34 nanometer or 3.4 angstrom unit. So these are the features of double stranded DNA or the double stranded DNA structure proposed by Watson and Francis Crick. We will stop here. We will discuss the next point in tomorrow's lecture.